All right, well, good afternoon, everyone, and welcome to our next series of Lunch and Learn at Cure Cancer. And we're delighted to have Marjan Naein here today. Thank you to all our observers and board members and team members that have turned up. I hope you find this session an interesting session of discovery, learning more about Marjan herself and also her work as well. Just a bit of housekeeping. If you don't mind, please just check that you're all on mute. If you do have questions, and we absolutely welcome questions, please pop them in the chat, notate them to me, and then I will fill them in as we have the discussion with Marjan. And this session will be about 40 to 45 minutes long. So we're delighted to have you here today, Marjan. This is the first time we've funded you, so welcome to the Cure Cancer family. Marjan is funded through the Priority-Driven Collaborative Cancer Research Scheme with Cancer Australia, and she's at the Garvan Institute of Medical Research, looking at esophageal tumours. So we're very much looking forward to hearing about you and your work. Thank you for coming today. Before we kick off about research, do you want to introduce yourself and talk about a bit more about how you got to this stage in your life?
Yes, sure. Thank you, Nikki. It's my pleasure today, and thank you for your invitation. So, hello everyone. I'm Marjan Naein, and I'm a computational biologist at Garvan Institute. I had undergrad genetics and human genetics, and I was like a vet lab person. And I just wanted to introduce myself in more like a informal way. And I was a vet lab person and human geneticist, which was interested to the genetic differences for the complex trait, including like cancer. And it was really challenging for me because I think I love challenges and I love solving problems. And that's why actually I was interested to this area. And well, out of like all the complex traits, I think cancer is really complex. So this is one of the reasons that I actually chose the cancer research area. But after that, the new technologies come in, like the big data and the genetic data. And then I realized, oh, I really need the computational skills to understand it pretty well. And that's why I did a PhD in computational and systems biology and Genome Institute of Singapore, back in Singapore. And I was graduated in 2018. And then I started a postdoc in Brisbane, Queensland, Australia at Kiwayama-Beckhofer. And I was there for a postdoctoral researcher focusing on the esophageal cancer for 3 years. And I think I'm really fascinated because it's really complex. And at the same time, I think there's a lot of patients still needs care. So I thought that probably this is the way that I have to do. So still continuing.
Oh, brilliant. Well, we've got something in common. I've spent 3 years in Singapore myself. So there you go.
Oh, nice.
You're a bioinformatician or a computer scientist, as we call them. At Cure Cancer, it is one of the specialities that we like to fund, and we're fortunate we're funding 3 of you this year. Could you talk a bit more from a day-to-day perspective? How is it different from a normal researcher in the lab? What do you do on a day-to-day basis in your role?
Well, it's the thing is, our job is just in the office, and it's like just with a computer. So somehow it's good and somehow it's bad because you can do it everywhere. So you just do it with a computer and just like a link to the like high performance computing of the university or the company that you're working with. But it's hard because you're all the time sitting. And it's like, and you can do it everywhere. So it's really hard to do the life and work balancing. And, you're thinking all the way, all the time, and you can do it from everywhere. So I think this is the challenging one, but it's at the same time, it's really good. So both are really interesting, but it depends. I mean, I think myself, I'm better than like in thinking and like performing on the computer rather than like doing the real wet lab things. I think both are interesting and they are complementary to each other. So yeah, it's different, both are, but different.
Yeah, they are. We talked to Sandra Brosder, and she also does something similar, and she talked about how important it is to have the computer, the biologist or the bioinformatician in the research sector as well. So are you part of a larger team looking at your research, or are you working on your own at the moment?
Actually, I am part of the, well, in our work, it's like everyone's like part of the bigger group. So actually, I'm part of the data science pillar, the central part of the Garvan Institute. So apart from the project that we have for ourselves, for example, I'm working on the esophageal adenocarcinoma, and I am also working on the zero childhood cancer projects at the same time, but also we help other people like the wet lab people who doesn't have any computational skill to help them to learn or like do computational analysis for them. So yeah, actually I'm part of a bigger group. It's a data science pillar, and Sarah Kammerfeld, she's like the director. And yeah, so we have our own project, but also we help people in their projects as well.
Yeah, look, and the Garvan is a fantastic place to be at for that. I know when I first spoke to you, you were in the process of moving as well. So what brought you down to New South Wales and the Garvan?
Well, I all the time, well, QIMR was really awesome. And the reason actually I was in Brisbane was because of a personal reason. My husband, he was doing a PhD down there. But I was also very much interested in Garvan Institute because they have a lot of like the sequencing facilities. The sequencing means like they produce a lot of genetic data, and they're also pretty good people who are very expert in just computational skills. So I really wanted to experience this. That's why I moved down here. And yeah, it's more, I think it's more personal, but I think I was all the time wanted to do this. I think I really wanted to experience this environment as well, which they have different expertise and they are more expert in the computational aspect of it rather than like the genetic part of it.
Yeah, lovely, thank you. So your work is on esophageal cancer, or the part that we're funding for you. Could you explain a bit more in layman's terms about what you're doing around esophageal cancer tumors?
Of course, yes, sure. So for the past 3 years, for the past 3 years, I was working on the like the trial data. It was Dr. Trial AGITG, it was an Australasian gastrointestinal trial, and they were looking for like new adjuvant therapy to improve the patient's outcome. And I was working with like the clinical data and the very big genetic data as well. And well, I found a group of people who unfortunately did not respond at all to any kind of treatments. And apparently it's not only because of like the treatment by itself, but it seems like the tumor, they have some immune features that like we call it like immune suppressive tumors. This is not about the immunity of the body, but this is about the immunity of the tumor by itself, which like tumor cells like bring a structure that they doesn't allow immune cells to kill the cancer cells. So it's really hard to tackle this problem. And we thought about it, all right, so we need to find a new therapeutic option for these group of people who have this problem and they are very like poor outcome. So, well, because we did a lot of like analysis, we found some features which was really complex, genetic complex like features which we couldn't really like solve the puzzle previously with the current technologies, and it's very complex. And this kind of, we call it like the structural variants, which the genomes is really messy. So as you know, that every day a new technology coming, and it's called like, we call it long read sequencing technologies that Oxford actually like announced it. So what we want to do, we want to apply this technology on this group of people. And we hope that we can find novel mutations and novel therapeutic like target for future that we can like like provide some kind of treatment for these group of people.
So when you say that they don't have, or the cells there don't make a change, or they're not with the current treatment, they don't work, what's the current treatment that these patients are going through at the moment that the cells aren't responding to?
Previously, during the past couple of years, most of these patients, they just went through the direct surgery. But thanks to the clinical trials recently, they think that pre-operative chemo and radiotherapy can help like a couple of people. Also, there is a couple of like other like treatments for other cancer type, like immunotherapy and like the pathway-based treatments, which unfortunately doesn't really work on the esophageal cancer. It improved like around 20 up to like 30% of the patients. But unfortunately, the esophageal cancer is really heterogeneous. It means people are very different, tumors have different features. So what we are trying to do, we are trying to like stratify or make patients in a groups and like support the treatments based on their tumor features rather than like applying one cure for all. So this is what we're trying to do. And we think that there is one group of patients that unfortunately we don't have any treatment option for them. So we hope that we can find this treatment option for them in the near future.
No, that's great. Thank you for explaining that. So just help us understand. So the esophagus is quite a long body part. Sandra was talking about how she's focused on the end piece of it, which is, and often there's a lot of cancer caused by acid reflux. Are you looking at a particular part of the esophagus or all of it? And it's just purely about patients that aren't responsive to current treatment?
So no, actually what I was working on, it was like I was working on the upper part, middle part, and the like lower part. And it is actually very interesting that these group of patients and people that I found that they're not responding pretty well to the treatment. It's not belong to the specific group. So it comes from like the upper, lower, and middle. So this is why we think that it's important, because it like covers more number of patients that we can help.
Yeah, that makes sense. Thank you. Okay. And then we also talked to her about the type of the patient that she was seeing in her analysis, and she was saying for the lower piece that she's looking at, it's 80% male samples. Is there a weighting that you can see for male versus female in what you're doing as well?
Yeah, so that's the thing. I mean, I think most of the esophageal cancer patients are male.
Yeah.
Well, I'm not very much sure about the epidemiologic reason and why it's like that. But yeah, but what we found is like even 90% of the patients were male, and we don't know really why.
Interesting. Okay, thank you. So these are the patients that are not responding to any treatment at the moment. Yeah. So you've got all this data. What do you do next? What do you analyze? What findings do you find? What are you looking for to look at features, trends, markers that you think might help improve their situation?
Yes. So the thing is, the thing what I'm actually working, I'm not very much sure if you're familiar with whole genome sequencing. So this is what we are doing. Previously, all the research at this point are based on so-called short read sequencing technologies. So what it does, actually, it's chop the sequencing to small reads and then they sequence it. But the newer technology that we want to do is like they cover a longer part of the reads. So why it is important? Because sometimes we have some kind of mutations that one part of the very important gene that we call it driver gene because it's important in terms of like starting the cancer or progression, and it moves to somewhere we cannot track it where it went and what combination it will make with a new gene with the new capabilities that we can like target and, for example, and tackle the problem. But with this new technology, we can track it down where it goes, and hopefully we can find like a novel mutation we can like find a therapeutic option for. So that's why this is important, and that is why actually what we do. It's kind of a like we have some like we found already some puzzles in the esophageal cancer that they improve the outcome, but I think with adopting new technologies we can tackle these complex problems in terms of the genome, because it's like the esophageal cancer genome, if you look at it, it's really messed up, and they break and reassemble together. Yeah. And that's why it's really hard with current technology. We follow up all the events in there.
That was really interesting to me when Sandra was saying that as well, that because it's heterogeneous, that it changes completely at different stages according to the various different treatments you've had. So you've got some cells changing quickly, some not changing, and the esophagus apparently is one of these places where it's well known for doing so, which makes detection of and treatment of this cancer even harder.
Yep, that's right. Yeah. So so it's and also some of them some of the events in the esophageal cancer is very interesting, because some of the events it happens at once. It's like the sudden changes, and then there are some that they're accumulating during these stages. So yeah, so it's really heterogeneous, and I think one of the important things that we can do is like to stratify patients based on their tumor features, and we tackle the cancer.
Yeah, lovely. And so you said that this long sequencing approach has come from Oxford University. Is that right? Yes. So how new is this approach?
Well, the approach is it's been a while, that like 5 or 6 years that's been around, but the first technology that it was introduced, it wasn't so great. It happens with all the technology, and it takes a couple of time to be improved and be ready to adopt it into the real science. So it's been a while, but I think it's the time that actually the genetics is revolutionizing, and they're going to replace the current technologies with they're calling short read sequencing. And why it is important, because we need to understand the usage of it, and whether we can, well, for example, in this project, before we going through a very big project, it's important that we understand the usage and whether we can find the novel things with this technology or not really. So this is the thing. But I think based on what the pilot studies have been done, especially in terms of the rearrangements, it's doing great. It's really great, and it's replacing the current technologies at this point.
Well, that's exciting, isn't it? You're leading the way. So you're looking for genetic mutations. I've got a question here about sample size. So what volume of samples or patients are you looking at the moment?
The thing is, well, previously when, if you remember, 20 years or 15 years back, when the sequencing came around, it was so expensive. But gradually, with time, all the time, the costs are falling. And again, for this technology, unfortunately, the technology is really expensive. So at this point, we can just look at 20 samples, because for each, it's 20 samples, but we call it 10 normal and human match pair. Mm-hmm. And that's why for each pair, it costs around $8,000.
Wow.
It's so expensive, but hopefully the prices are falling during the time in future.
Yeah, I'm sure as it becomes more common as well. So you're looking at these mutations, and you're trying to see if there's a link. You identify that, then what's next? What's the kind of the next stage in the research?
So, well, this is very important, because we get the understanding of, for example, cancer and these group of patients, and they have these features. So the next step would be because we have clinical collaborators. So sometimes, for example, when they affect specific genes, they already have some kind of treatments or drug. So they can like use it as a clinical trial. We can like help them, but probably this translational part, it would be just like adopted to the clinical trials, and they say whether these group of patients are they really like they can be treated with these or not, and then if yes, they're going to be like a real treatment. But it's very interesting because we actually, for these group of patients that I'm talking about, and we call it immune suppressive groups, we did actually a lot of works, and I think in another project also we are actually trying to make an IHC test, immunohistochemistry tests, that we can like with a biopsy we can like distinguish them from other patients. So we did that already.
Wow.
So the thing is, now what's the next? So what would be the potential therapeutic? So at this point, anything we find would be potential, and hopefully if we can like publish it in a like good journals, then the clinical part is ours, and other people, they can work on it and introduce other like clinical trials.
Yeah, wonderful. Thank you. So from your perspective, what's your hope? Obviously, you just talked about tests that you've helped establish there at a clinical level. In this project, what's the end result for you? What's good progress?
Well, the good progress, I think we have a pretty safe progress. The thing is, because we, the project, when we started, we actually searched a lot of aspects of the esophageal cancer, and we know that this is like, and these complex events has been reported not only by us, it's been reported in many journals like Nature by itself, Nature Genetics, and it's there and it's real, and we don't have any like solution for it. And definitely the progress would be that we can like find a novel mutation with these technologies from these patients. So I think this is a good progress, and we can really find something very like we can resolve some of the complexities. Yeah,
it must be hard, because in fact you probably don't know what you're looking for until you find it as well. So it's kind of a catch-22, isn't it, that you're looking for some mutations, but then you might find something else that takes you down another path of your analysis.
Yes, exactly. But anyway, I think applying long read sequencing is these days it's a very hot topic, and it's very important because the science is gradually going toward that way.
Yeah.
And even if we find like some mutations, or we can understand more about the esophageal cancer, but we are also thinking about the national, we can like establish ourselves as a national people, even international, who are experienced working with long read sequencing, and not only in this cancer type, but also other cancer type, that we can help to find novel mutations.
Yeah, look, that was actually going to be my next question with this long read sequencing. So you're doing it now for esophageal cancer. I presume it can be used for many other cancer types as well.
Yes, actually, it's not only esophageal cancer that it hits by these kind of complexities, and we call it structural variants and these structural rearrangements. There are other cancer types like melanoma. That's one of them, and also like neuroblastoma. There's a couple of cancer types that are hit by multiple structural variants, and it's kind of a mutation. It's not a like point mutation. It's a mutation that covers a region. So it's very important. And to be honest, I actually had a grant from the Australia Skin Cancer Society. And previously I was working on the acral melanoma, which is very important. Like structural variants are very important in this cancer type as well. And we actually found a novel mutation that we couldn't find it previously. And when we compare it with the short read sequencing, it was very mixed up but we could see it clearly with the long read sequencing. So it's very important because we could track where it went.
Yeah.
We could track it down. So but melanoma is slightly easier to work with compared to I don't know, carcinoma.
Yes, OAC is a tricky one.
Yes.
So that, from a career perspective as well, for you, that must be fascinating that you can look across several cancer types too.
It is, yes. Well, actually, during my PhD, I was working on 21 cancer types.
Oh, wow.
Yes, I'm experienced in working with different cancer types. Yes, but it's very important that, you know, when I started to looking at like the cancer in a very early stage as well, I was undergrad, I thought, oh, maybe we can have a cure for all, but it's impossible because I think each cancer type has their own features. It's like different humans. So yeah, so you have to like understand them and respond based on their features. So this is what we are like considering for each cancer type, and also not for each cancer types, but for, we have to like stratify each cancer type to different subtypes and like treat them differently.
Yeah, it's very complex, isn't it?
Yeah.
So let's just talk about your funding. Obviously, this is the first time we've funded you, and like many researchers, you spend a lot of time applying for grants, I am sure. What does this early career research funding mean for you?
Well, I think it means a lot for me because I'm a person who loves science, and I was thinking all the time, and I really want to apply these problem solving on the current problems and really want to help cancer patients. But unfortunately, research is really expensive, and you're not able to do research or make resources without money. So this is really important from aspects of research. But also not just from me. I know many talented early career that they're really talented, and I'm pretty sure they can help like maybe like introduce new biomarker or new treatment in the future. But unfortunately, because they don't have the like fundings, they're not able to apply their thoughts. So yes, I think it's very important. And besides that, we consider that early career have to all the time compete with mid-careers for the grants, and it's really hard, and it's, well, there's track records is not the same. So it is a way that they can establish themselves as a like researcher and become independent gradually in research. So for me, it's personally, it helps me with a career, but also I can like apply my own ideas to help cancer patients.
And on that, what's the most rewarding part of your job? What's, you know, what gets you up in the morning and makes you excited about doing your work?
Well, I really love science, and this is like the well, I love challenging part of it. And as I mentioned, like our projects and what we work, the hard part is, well, sometimes it takes a while, maybe years, to see the outcome. So you should be patient, but at the same time, you have everyday question, what you want answer. And sometimes you, for example, you see something, you want to know whether it's true or not. So you just get up and start working. And yeah, so these problem solving, these are all really fascinating. And I really love science. I think if somebody doesn't really like this solve problem, shouldn't be a researcher.
I've got a question here about how you keep your analytic skills and analysis up to speed. Do you have to do other courses on refreshing new technology? Is it the same as in the business world where you go to training courses and you learn more? Do you do the same in your job as well?
Yes, well, to some extent, yes, and to some extent, no. The thing is, we have different levels of bioinformation and computational scientists. Some people are people who just use and apply current technologies, current softwares or current tools for the technologies. But some computational scientists, there are people who are actually they write that application themselves as well. So yes, of course, we need to like because the genetic data is sometimes is that much big that you cannot. It's not like Excel and you can open it. Sometimes it's like it's very big. It's like gigabytes that you cannot even see what's going on there. So for doing the analysis, you all the time need to know the faster ways, like the maybe scripting languages, or for example, recently this Google, for example, clouding languages comes around, Amazon is coming around. So these are the things that you can help yourself and do a faster or improve analysis. But in terms of doing analysis, for example, for the long read sequencing, it's pretty new technology. So some people, especially we have a lot of people at Garvan Institute, that we are actually making our own tools, which actually will provide other researchers to actually use it. So yeah, so yes and no.
Well, I have a question here from our analyst, so it is rather analytical. He wants to know what's your main language that you work with? Is it Ruby or Python or something else?
Well, yes, it's a really good question. It very depends on the level that we are working. So I'm a shell R scripter and Python scripter. So I use, and also sometimes use MATLAB, because it very depends on the, if there is a tool that package is also, for example, only on MATLAB, I have to use it, I'll use it. In the Python, it very depends on it. But for example, in a very early stages of the data analysis, especially whole genome, they're mainly we are doing in the shell, and we call it some kind of workflow languages. I don't know, we call it WDL, WDL. So they call it workflow languages, but they are mainly shell scripting. But when as soon as you have like your clean data, which is summarized, we bring it to R or Python for further and downstream analysis.
Lovely. All right. Thank you. Thank you for answering that one. Another question here about your international collaborations as well. So you talked about how Oxford University implemented this long sequencing that you are now working with. Do you currently work with other teams from elsewhere in the world on what you're doing, or is it Australian based at the moment?
For these projects, actually, it's Australian based because the interesting part is the Garvan Institute is the second center after Oxford, is the first actually center that has a long read sequencing. And I believe that the computational scientists that we have at Garvan Institute, they are pretty expert in it. So yeah, so we have actually a very good center to perform this. So no, at this point, we don't have any international collaborators.
Doing it yourself, and that makes sense. And then a question on that. So obviously you come down to the Garvan, it's world renowned for its research and analytics. And, I know that they look after data sets and copyright that as well. That's how advanced the Garvan are. What's for your future? Where do you want to be? If we fast forward your career in 5 or 10 years' time, where would you like to be?
Well, in terms of in terms of research, I really want to continue what I'm doing, because I think this is an endless job to working with data, clinical data, and also find biomarkers, therapeutic target, and never ends. You just like feel some like missing puzzles, but I think it never ends. And all the time you can help it till have a world without any cancer. On that time, I don't have any job.
Sadly, it might be a while.
That's a good thing.
Oh.
But for this purpose, I really want to become an independent researcher gradually. I definitely would like to apply for the investigator grant next year. And I think this grant is really helping me out to establish myself and my skills and my ability that I'm able to do this. And yes, so gradually getting independent, having my own lab, and going higher and higher in independencies.
Oh, brilliant. Thank you. And our final question, obviously we've got our board observers, team members here. What else can we do to help you?
Well, I think first I have to thank you for this opportunity, because I think this is really important. That the funding that you actually this opportunity that you gave me. I think I don't really have anything in this moment, but maybe with getting the direction, what is important for the cancer patients, and how we can like synchronize our work with the consumer's needs. I think this would be really great.
Yeah, look, well, thank you. Thank you for your insight. It's been fascinating to learn a bit more about what you're doing, and how exciting for you that you're doing world-class research behind Oxford University as well. It's fabulous, and it must be very rewarding, although challenging, I can imagine. So thank you for coming along today. It's been a pleasure to talk to you. Thank you to all our participants as well. And I look forward to welcoming you all back next week, where we'll be chatting to Dr. Marjan about her work on adult brain cancer. Thank you very much, and have a good afternoon. Thank you.
Thank
Bye-bye.
you. Bye.